Hi everyone, this is Sandy Cottle. Today's lesson is going to be out of Chapter 2 in your textbook, and this is the importance of organic chemical compounds. We're going to be talking about how chemistry comes alive in biochemistry and how important these compounds are in our body. You'll probably already know this, but we don't use every topic out of the first few chapters in the first unit. We select them according to when we want to introduce them, so you won't be getting everything out of Chapter 2. But anyway, without further ado, let's talk about our organic compounds. There are 4 basic organic compounds. They are carbohydrates, lipids, proteins, and nucleic acids. One of the things that they have in common is that they all contain carbon. Most biologists would say that carbon dioxide and carbon monoxide, these two here, even though they have carbon, are not traditionally thought of as organic compounds. Now, organic chemists might disagree simply because they do have carbon in them, and that's one of the main determinants. But we think of these two molecules as being inorganic. They're not part of these 4 major groups. So let's go. We're going to discuss each individual group and talk about their structural units. So the first one we're going to address are the carbohydrates. When we think of carbohydrates, I am sure that you recognize them as things like our sugars and our starches. The first thing we'll do with each one of these is we'll talk about their basic structural unit. What is the smallest unit of structure and that has a specific function that gives carbohydrates their unique properties? So the first one of the carbohydrate's smallest functional unit is called a monosaccharide. You need to recognize some of the pieces and parts of the word. So the first thing I want to talk about is mono just means what? You probably already know that. One, right? One. Let me see if I can make this line a little bit bigger. Okay. And sacca in Latin means sugar. So the smallest functional unit of a carbohydrate is a monosaccharide, or a simple sugar, 1 unit. The most common one that I'm sure all of you all have heard about is glucose. But glucose is the most common 6-carbon sugar. Notice down here that all carbohydrates have carbon. They also are going to have hydrogen and oxygen in them. The other monosaccharides that are 6-carbon sugars are fructose and galactose. But then we've got a couple of very important sugars that only have 5 carbons in them that also are monosaccharides. The 5-carbon sugars are ribose and deoxyribose. I'm sure you've heard of those before too if you've had any biology at all, because these are the 2 types of sugars that are found in DNA and RNA, and we're going to talk more about those later. Alright, so carbohydrates then are built from these basic building blocks, or monosaccharides. When you put 2 or more together, 2 sugars or more, they are considered disaccharides. So di is 2, and saccha, of course, is sugar. The most common disaccharide is sucrose, and that's what you put on your cereal in the morning, probably. It's called table sugar. And sucrose is actually the two monosaccharides that go together are glucose and fructose. So you put these 2 together, chemically bind them together, and you get the molecule we call sucrose, the disaccharide sucrose. Other disaccharides that are real commonly seen, especially in the body, are lactose. Lactose, that doesn't look much like a C. Lactose, do you know where lactose is found? Lact is a root in Latin and means milk, so this is sugar. And also notice how all of these have an ending OSE. When you see OSE, I want you to think sugar, okay? OSE means sugar. So lactose is milk sugar, and another real common disaccharide is called maltose. So it is malt sugar. You would see that in some of your candy bars. If you look at the ingredients on, like Milk Duds, I think, have maltose sugar in them. So anyway, next time you're at the movies and buying the candy, or at the grocery store and buying the candy, take a look at the ingredients, see if you recognize some of these. Then we can also take sugars and we can arrange them into polysaccharides. What would poly mean? Many, right? If you take many single monosaccharides and hook them together chemically, and you would get the most common polysaccharide, which is called glycogen, and it is a form of animal starch. When we talk about polysaccharides in plant material, we just use the word starch. But typically when we say starch, we're talking about polysaccharides in plants. When we say glycogen, we're talking about polysaccharides in animals. And glycogen is the most common form in the body to store a sugar in. So the liver actually can hook a bunch of simple sugars together and store them as a glycogen somewhere in our body. All right, so back to emphasize, the main elements found in carbohydrates are carbons, hydrogens, and oxygens. And we often abbreviate carbohydrate as CHO, like that's carbohydrates. It's actually just got the symbol for each of the elements that are found in it. What do we use carbohydrates for? I'm sure most all of y'all know that our carb is where we get our energy from. And you're going to see that it's used, carbohydrates, as they're broken down and absorbed into the bloodstream and then get into our cells and go through a process called cell respiration. Our carbons are basically broken apart. The bonds that are between each of the carbons are broken apart, and that energy is transferred that held the carbon bonds together is transferred into what we call a phosphate bond to create the most usable form of energy by cells, which is ATP, or adenosine triphosphate. The very last couple of slides today are going to address ATP in a general way, so we'll get back to that. Another thing that carbohydrates do is carbohydrates sometimes are used to form structures. For instance, we just mentioned that ribose and deoxyribose were sugars that were a part of DNA and RNA. So one example of a structural carbohydrate is the sugars used to form DNA and RNA. Another example of structural carbohydrates are glycoproteins. And glycoproteins just mean that you take a starch molecule, a polysaccharide, and attach it to a protein. And where we often see these as a part of structural proteins in our body is they're expressed on the cell membrane or made by the cell and put out there on the cell membrane, and they form what we can sometimes call markers. It's like, this is what I am, I'm a muscle cell, I'm going to need a lot of sugar, or whatever. So the markers are kind of road signs, if you will, that tell hormones and so forth, where to go, what to bind to, and things like that. You'll learn a lot more about these things later. All right, let's go to our next one. The next organic compound we're going to talk about are lipids. They're distinguished primarily because they are not water soluble. We sometimes say they're hydrophobic, which means they fear water. Okay, so what they will dissolve in is they will dissolve in other fats and organic solvents. Kind of like when you use the oil-based paint in your home. You know, you can't clean an oil-based paint with water. You have to use paint thinner or turpentine or something like that. Those are examples of organic solvents. The most common dietary lipids are triglycerides. That's what we call them. These are the most common dietary, what we bring in our diet. And these are made up of fatty acids and a molecule called glycerol. Fatty acids are often abbreviated in biology as just little f, little a. And fatty acids are often described as hydrocarbon chains. And basically, so all they have is a chain of carbons with a bunch of hydrogens around them. Notice how carbon has 4 bonds. I'm not sure whether this is showing up very good, but, you know, carbon can bind with 4 different things. And so you have fatty acid chains because they have long carbon backbones with hydrogen around them. But they also have, attached to these, is a glycerol. I want you to recognize the OL ending. Anytime you see an OL ending, I want you to think alcohol. What is an alcohol? An alcohol is just a fermented sugar. You're going to learn more about respiration and how we have aerobic and anaerobic respiration. But anaerobic respiration is how we make, how we ferment a sugar and make alcohol. It's just like you ferment the grapes to make the wine, okay? So a triglyceride, most common dietary fat, is made up of a glycerol molecule with 3 fatty acid chains, and that's why you get the triglycerides on there. So it's a glycerol molecule, a fermented sugar, which is an alcohol, with 3 of these long hydrocarbon chains attached to it. These form things in our body, fats, oils, and waxes. I know you know what fat is, so we'll skip that. Oils in our body are like sebum. You know, sebaceous glands secrete oil that makes our hair kind of oily and greasy. Another place that you would find a wax would be on the surface of our skin. We have a protein called keratin that is actually a waxy protein. It is a lipoprotein, or a lipid combined with a protein that waterproofs our skin. So you think of wax having that kind of a property. So we'll see how that is produced on the surface of our skin to waterproof our skin. Not just to keep water from coming in, but more important, to keep water from leaving our body. Where else do we see lipids in the body? One of the biggest makeups is in the cell membrane. The cell membrane is described as a phospholipid bilayer, and you will study that in this unit. You'll have a whole video on the cell membrane, so you should recognize a phospholipid. When you see it, it's just a fat molecule, a lipid with a PO4 phosphate on it. A phosphorus with 4 oxygen molecules attached to the lipids in that part of your cell membrane. Also, important lipids in our body form steroid-based compounds. An example of a steroid-based compound that is important would be, say, certain hormones are steroid-based, and particularly some that you would be familiar with that we refer to as the sex hormones. Can you name the sex hormones? What's the male sex hormone? Testosterone, of course, and the most common female is estrogen. Now, let's go back and look at the elements that are found in lipids. Notice how there's exactly the same as found in carbs. How do you know the difference between a chemical compound that they all have carbons, hydrogens, and oxygens? Are they a carbohydrate or are they lipids? Well, chemically, the biggest difference is the fact that there are fewer oxygen molecules in lipids than there are in carbohydrates. And if you had time to take chemistry, you'd learn all about that. So let's go back and sum up the functions of lipids. Lipids act as energy stores, primarily, number 1. They also act as lubricants in our body. They form cell membranes in our body. They form certain hormones in our body. And the fat actually is involved in insulation. I didn't put that on here, but we'll write it on here now. So, insulation in our body. All right, let's talk about the next group. The next group are the proteins. The smallest structural unit of a protein is an amino acid. We often abbreviate that little a. This is the smallest unit of a protein that exhibits the properties of a protein. There are 20 common amino acids found in the human body. You can look them up in the textbook if you'd like to know their names. They're often listed there. There are 8 of these that are called essential. What that means is there are 8 that you absolutely have to have in your diet. The rest of the 20 that are used in our body to make various proteins can actually be made by rearranging the molecules in the liver. The liver can do it. But you absolutely have to have the 8 essential amino acids in your diet for your liver to be able to rearrange things or metabolize them and to form the rest of the amino acids we need. Peptides are where you have 2 or more amino acids linked together, bonded together. What would a dipeptide be? Di means what? 2. So 2 amino acids put together form a dipeptide. What would a tripeptide be? Where you've got 3 amino acids put together. So it's as simple as that. And then when you get more than that, we tend to get into what use the term polypeptide. Of course, poly just means many again. Most of our polypeptides are pretty long and rather complex chains, meaning they have lots of carbons with lots of molecules around them, tend to have at least 50 or more amino acids. Once polypeptides get to be a very specific structure, certain amino acid first, second, third, and then they start twisting and turning and folding and forming things. When they get to the point where they actually form a specific function, we call them a protein, and then each protein has a specific name. For example, one of the hormones that's a protein-based hormone you'll be studying is insulin. And insulin is one of the most simple of all of the protein hormones in the body. It is only 51 amino acids long. But once you have the very specific sequence of these amino acids and you form the protein we call insulin, then it will have binding sites that allows it to do very specific things. And I'm sure you know already the main function of insulin is when it binds to certain cell membranes, it allows that cell to transport sugar into the cell, and what that does is lower your blood sugar. So I know you know that insulin lowers blood sugar, but that's how it's done. There are lots of proteins we're going to be studying in the body. In the next unit, we'll particularly be introducing things like collagen and keratin, and after that we'll be talking about the muscle proteins, actin and myosin, so you'll be getting a lot of these. In fact, I have some of them listed down here, so we'll get to that in a minute. All right, notice how the proteins have the same 3 basic elements that the first 2 molecules had, carbons, hydrogens, and oxygens. But now notice something. Now, proteins would not be proteins without nitrogens in them. So one of the things that makes them unique is the fact that they have nitrogens. You probably know that a common nitrogen compound in your kitchen or in your closet, your cleaning closet, I can't even write, is NH4. You know what that is? Ammonia. Let me give you just an example. We're going to be talking about how the kidney, one of the kidney's functions is to filter the blood of nitrogenous waste. So any compound that's got a nitrogen in it, the kidney's going to be filtering some of the excess nitrogens out. That's the reason why our urine smells like ammonia, because it has the properties of ammonia. It has nitrogen and some hydrogens on it, and that makes ammonia, and it smells that way. So, also, some of the proteins will contain sulfur, and some of them will contain phosphorus. Sometimes to help students remember this, I tell them to remember it like this, CHNOPS. If you do it that way, you can pronounce it schnapps. So if you want to remember all the elements that are often found in proteins, go carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. And I think that's actually even in order based on which of the elements are in the most prevalent. But I may be wrong about that, so don't hold me to that. Okay, so what are the functions of proteins? Proteins form building blocks. They form fibers or structural proteins. So when we say fibrous, think of fibers. I know you've heard about collagen. It's the most abundant protein in the body. Collagen are fibers that make our skin, strong. It adds strength to our skin. And you'll learn of other fibrous proteins like elastin that allows our skin to stretch and then pop back to its original tightness. As we age, those proteins start breaking down, so you lose the elasticity produced by the elastic. But these are called fibrous proteins. They are building blocks. Other proteins that form building blocks are the proteins that allow muscles to contract. And you know how muscles will get bigger as you work out more, as you require or demand them to do more action. The muscles actually hypertrophy or get bigger. And how that happens is that every cell in your muscle is trying to respond to this demand, and it's actually going through the protein synthesis to make more of the contractile proteins like actin and myosin, and you'll learn some others that allow you to make these structural units that allow the muscles to contract. And so you're adding more proteins inside every cell, and the cells are going to get bigger. Now, so they act as building blocks. They also can be globular, and when they are globular, they are functional proteins, not structural proteins. That means globular proteins have binding sites. You will have a whole video, I think, in the next unit on how how proteins are organized that allows them to have binding sites. But anyway, you'll learn more about that. Globular proteins have binding sites here, and because of those binding sites, they form enzymes. They form hormones, certain hormones. They form molecules like antibodies. Don't know what just happened there. See if I can back it up. Okay. See if I don't have my pen anymore. Another red. Anyway, antibodies that function in our immune system are protein-based and have binding sites on them. So anyway, proteins are going to be so important. You're going to learn so much more about those. We're going to go on to the fourth group, which are the nucleic acids, and I'm sure you have heard of DNA and RNA. Deoxyribonucleic acid and ribonucleic acid. The smallest structural unit is called a nucleotide. A nucleotide contains phosphates, which are PO4 groups, ribose sugars, remember those are your 5-carbon sugars, and then they contain bases that I like to call nitrogenous bases. That helps remember that nucleic acids also have nitrogen in them. The nitrogenous bases are adenine, thymine, guanine, and cytosine that are forming DNA. DNA down here functions as our genetic material, so our genes are stored in DNA. And these typically bind very specifically. A's bind to T's, and G's bind to C's, and they will form that ladder-like thing that is twisted. We call a helix, a double-stranded molecule that is twisted that you recognize as DNA molecule. But what holds that ladder together are the nitrogenous bases, adenine, thymine, guanine, and uracil. When you're looking at RNA, the nucleic acid RNA, notice it has adenine, uracil, guanine, and cytosine. What happens there is RNA does not use thymine. RNA substitutes the molecule uracil for thymine. So in RNA, you will have A's pairing to U's, but G's will still pair to C's. RNA's main function is in protein synthesis. So when you think RNA, I want you to think protein synthesis. Let's see. Okay, so let's go back here and say one more thing here. Notice that these contain carbon, hydrogen, nitrogen, oxygen, and phosphorus. So SNOP or nucleic acids. So I remind you that this one also contains nitrogens. When you talk about the function of the kidney, we're going to be talking about the kidney is removing nitrogenous waste as well as some other stuff, but you should know that that waste is coming from proteins, which have nitrogens, and nucleic acids, which have nitrogens. When you look at RNA functions, I want you to think protein synthesis, and you will learn more about this, but RNA comes in three main forms. Little m, RNA, which stands for messenger, little r, RNA, which stands for ribosomal RNA, and then you'll have a tRNA, which is transfer RNA. How do DNA and RNA differ in their functions? Well, DNA, of course, is guiding cell division, or cell replication, let's call it like that. So when a cell mitotically divides to form a new cell, we call that something the DNA is important to do. You've got to replicate your DNA in order to create a daughter cell that looks just like the mother cell. Another thing that's important is that DNA does. The genes for DNA actually determine the amino acid sequence for each protein. So you wouldn't know how to make your proteins if you didn't have the specific coding on, and I'm talking about coding, a code. A triplet code would be a sequence of three codes that would tell it what amino acid should be used to make each of the proteins and in what sequence they're used. I just had a thought when I looked down. I want to back up a second. I'm going to go back, if it'll let me go back to proteins, with something important that I wanted to say that I left out about proteins. So we're backing up here. I wanted to use an example of a globular protein. One of the most common examples of a globular protein that you've heard about is hemoglobin. Notice how even GLOB is a part of the word. That tells you it's a large protein, globular, like a big glob, and when you get globular proteins, you're going to have binding sites. What does hemoglobin have binding sites for? Sure, you know that. Oxygen, it even has binding sites for carbon dioxide. Another thing I wanted to mention about proteins, and I'll write it up here, is that proteins can't work if they have been denatured. And you can denature, which is to totally change their shape, which knocks out your binding sites, which determines the function. Proteins are denatured by extremes in temperature or pH. And when you have denatured a protein, you totally prevent them from having the properties. A good example of that would be the white of an egg. You know, a raw egg white is clear and jelly-like, but when you subject it, that's actually a protein. It's a protein we call albumin. But when you subject that to the extreme temperatures of like putting it in a skillet and frying it, what happens to that protein, albumin, that makes the egg white? It turns white, doesn't it? And it's opaque, and it goes from a semi-solid to a solid. And can you reverse this? No. Once you have subjected that protein to this high temperature, you have forever changed its consistency. It's not reversible. That's the kind of thing that can happen in our body if our temperature gets too high. With a really high fever for an extended period of time, the proteins that function as hormones and enzymes and antibodies and a lot of stuff in our body will be denatured, and then they can't function, and that can be life-threatening for you. So let me see. I'm going to zip on here to the last slide, last topic. I told you I wanted to come back to adenosine triphosphate. So here we are. ATP, such an important organic molecule we're going to be talking about, because ATP is the immediate way cells need energy. It has to be in this form in order to do cellular work. An example would be when muscle cells contract. We know that muscle contraction requires tons of energy. Well, one of the places that it's used is on what we call the myosin heads, which is a protein. When ATP is bound to the myosin head and it's enzymatically broken up into its subunits, a DP and a free phosphate, you're going to have some energy released. And when that energy is released, that's what's going to allow the myosin heads to move, to change position. And as they do, they're going to reach up and grab actin and pull inward. And when they do that, the muscle actually contracts. You'll learn more about that later, so don't worry too much about what I just said. How do we get ATP? We've talked about how glucose is our primary energy source, and all of our carbohydrates are energy source. But that is absolutely useless until we have catabolized the glucose, which is to tear it down, to take it apart, in a process we call cell respiration. You're going to have a whole video on this. But that will ultimately lead to the creation, or by transferring the energy from the carbon-to-carbon bonds in the sugar molecules into a phosphate bond. You're just taking the energy and using it to attach phosphate to make ATP. In fact, the next slide is going to show you a little bit about that. And I'm not that interested in you learning this formula. I'm just using it to show you a structure. Let me get over here. We tend to think of ATP as we'll sometimes say that it's formed from this part of the molecule, which is referred to as adenosine. And adenosine is the nitrogenous base adenine, A-D-E-N-I-N-E, like the A in what? You know where I said DNA has an adenine that pairs with a thymine? It's the same molecule, adenine, plus a ribose sugar. That's what adenosine is. In this depiction here, this is your molecule that we call adenine. It is the nitrogenous base, adenine. This here is your 5-carbon sugar ribose. Every time you see a corner like this with nothing on it, it's understood in chemistry that that's where your carbons are. So there's 1, 2, 3, 4, 5 carbons in this ribose sugar. So adenosine, triphosphate, adenosine, that word is what we call in biology this combination of adenine plus ribose. Now, let's go over one unit here. Notice here is a phosphate molecule with 4 oxygens around it. We refer to that as a PO4. So an A plus 1 PO4, A meaning adenosine here, equals what? What's the word, the root word that means 1? There's several of them, but what would it be here? Adenosine plus 1 phosphate is what we call adenosine mono. Remember that root? Phosphate. Monophos. I can't spell them right sometimes. P-H-A-T-E. That's a P-H-O-P-A-S-P-H-A-T-E. Monophosphate. Then if you take and add another phosphate plus 1 more, so adenosine plus 2 PO4s is equal to adenosine what? What was the word that meant 2 a while ago? Diphosphate. Then when you add this third PO4 molecule here, you get adenosine plus 3 phosphate is equal to what? Adenosine triphosphate. Adenosine triphosphate. Now, I want you to notice how up to now, all the chemical bonds that you see everywhere are a straight line bond. But notice when we get here, you get what we call a wavy bond. You see these wavy bonds? Wavy bonds simply have a lot of energy in them. And these high energy phosphate bonds are where we hydrolyze them. We use water to break the phosphates off, and that's what releases the energy. And this is the form that the energy has to be in for cells to use it. To give you one more example of how much energy is there, I like to use an old playground model, if you will, an example. I can remember playing crack the whip when I was a kid. That's what we called it. My kids, when they played in elementary school, they called it snake, I think, or statue even. And it's where a bunch of people get in a big long line and hold on to each other, and you have a leader, and the leader starts pulling everybody around. Have you ever noticed how the longer the chain, the harder it is for the end guy to hang on, because the wave increases as it goes further down the line, and so that poor last guy is just slung all over the place, you know, and on the playground, you know, they land like a statue and stop. But anyway, what I want you to think about that with ATP is the shorter the molecule, the less energy. So it's harder for the leader to throw off the end guys as the line gets shorter. So there's less energy. So it takes a lot of energy for this third phosphate to hang on, because he's being slung all around. It takes a little bit less energy, but there's still a lot of energy in holding on the second phosphate. Once you break those two off, there's a little bit of energy left here for Na and P, but not enough to really drive cellular processes. So I hope you've learned a little something about organic compounds. You'll be hearing a lot more about those in every one of your videos. So I wish you well, and I'll see you on the next round. Bye. I'm trying to stop recording.